So good evening, students. So in the previous session, we started off with the topic called as pedigree analysis, right? So let's continue with the pedigree analysis. So we have been seeing what exactly is pedigree analysis and how exactly it is being plotted, right? So why do we go in for pedigree analysis? To monitor a particular character from generation to generation, right? So to monitor a particular character, how it is being transmitted, or rather how it is being inherited from one generation to the other generation, we usually go in for this pedigree analysis. So one of the major tools in human genetics, right? So we saw how exactly do we plot a pedigree when a particular question is given, right? So we have taken up a couple of examples with regards to that, right? So if a question is given, how do we plot a pedigree? So along with that, we have also seen that if a pedigree chart is given, how do we interpret the pedigree chart, right? So that was the things what we have taken up in the previous session. So with regards to how to interpret the pedigree chart, we have taken up a couple of examples. One, we studied something called as the autosomal dominant condition. And the second pedigree chart, what we took up was autosomal recessive condition, right? In this session, let us take up a few more examples and see how we can interpret the pedigree chart if at all it is being given, right? So remember the rules, what we said in the previous session. How do we identify a pedigree chart? First thing, as and when a pedigree chart is given, first identify whether it is a dominant condition or a recessive condition. So how do we identify whether it is dominant or recessive? By observing the affected individuals in the generations. If all the generations have at least one affected individual, then we say it is a dominant writer, right? If there is skipping of generation, right? If there is skipping of generation, we start saying that it is a recessive writer. So that's how we start eliminating whether it is dominant, or we start identifying whether it is a dominant trait or whether it is a recessive trait. Now, once we are clear with whether it is dominant or recessive, the next thing what we need to identify is whether it is autosomal disorder or a X-linked disorder, that is a sex-linked disorder. So how do we identify whether it is autosomal or X-linked disorder? First, look for the generations affected individual. If affected father has affected son, then it would be autosomal, right? Affected father having affected son, it would be autosomal, right? One more condition, what I said, affected father will have both affected son and affected daughter. This is another way how we re-verify whether we are going in a proper direction or not, right? If affected father has affected son, it is autosomal. Similarly, if affected father has affected daughter and a son together, then again it would be autosomal. The next thing, how do we identify whether it is X-linked disorder, right? In X-linked condition, affected father would have affected daughter, right? So if affected father has affected daughter, then that condition would be X-linked condition. And one more way, what did I say? How do we identify X-linked condition, right? If the number of male individuals affected are said to be more, then also we can say that it is X-linked, right? Why are male individuals affected more in X-linked? That also I told you in the previous session. Because especially in a recessive mutation condition, right? Especially in recessive mutation condition, affected males will be more than the affected females with regards to X-linked trait or X-linked character. So why that also I told you, right? Because in case of human females, there are two X chromosomes. If a particular disease has to be expressed, mutation has to be seen in both the conditions, both the genes present on both the chromosome. Whereas in case of males, since there is only one X chromosome, right? Because there is only one X chromosome, one single mutation would be enough for the expression of that particular disease, right? So this is how we start identifying whether it is a X-linked disorder or whether it is a autosomal disorder. So with regards to that, we have taken up two examples in the previous session. We saw how exactly a autosomal dominant pedigree looks like, and at the same time how a autosomal recessive pedigree looks like. So in this session, let us take up two more examples and see how we can interpret those particular pedigrees, right? So let us take up two more examples here. Right, so we are dealing with pedigree analysis. So we are dealing with the pedigree analysis here. So let us take up with the third chart. So we have taken up 2 charts in the previous session. So now let us take up with the third chart here. Right? So we have a male here. So the male is affected, right? Marries a normal woman, and they have 3 children. Female affected, second is a male, and third is also a female who is affected. This one marries a normal man, and they have 3 children. So this is the pedigree chart what we have. This is a 3-generation pedigree chart here what we have. Right? So this is the first generation, this is the second generation, and the third generation. Again, the options what we have here. The first option would be autosomal dominant. The second one would be autosomal recessive. The third one would be X-linked dominant. And the fourth one would be X-linked recessive. Right? So these are the 4 options what we have with regards to this particular pedigree chart. So you can just make a note of this pedigree chart. Right? So let us see what exactly is actually given in the question. In the first generation, there is an affected male. This affected male marries a normal female, and they have 3 children. The first daughter, second son, and third child is again a daughter. So both the daughters are actually affected. Right? In the second generation again, affected female marries a normal man. Right? Affected female marries a normal man, and again they have 3 children. All 3 are sons, and out of all the 3, 1 son is actually affected. This side, if you see again, the affected daughter marries again a normal male, and they have 4 children, 2 daughters and 2 sons. Both the sons are affected. So what type of disorder or what type of pedigree does this present? Right? So let us again start with what we have seen in the previous session. So how do we identify first? See whether this is whether it is a recessive condition or a dominant condition. So how do we check whether it is recessive or whether it is dominant? If I said, if all the generations, if there are affected individuals present, right? If in all the generations, the individuals are affected, then we said that it is a dominant trait. If there is skipping of generation, right? Just like what we saw in the previous session, if there is skipping of generation, if that is the case, we would confirm it as a recessive condition. Just observe here. So first generation, male is affected. Second generation, both the females are affected. Third generation, all the males are affected. Now, since in all the generations there are affected individuals being present, this type of condition would be a dominant condition. So our answer would be either option 1 or option 3. So option 2 and 4 are actually ruled out. Next, how do we identify whether it is autosomal or whether it is X-linked? Right? So what did I say with regards to autosomal? I said that affected father will have affected son. That is in case of autosomal. Right? Affected father will have affected son. Is that seen here? Affected father, do they have affected son? No, the son is unaffected. Again, father unaffected, the son is affected. So it cannot be autosomal. It should be X-linked recessive. So let us again reconfirm whether it is X-linked or not. So what did we say with regards to X-linked disorder? Affected father can have affected daughter, right? So here father is affected, both the daughters are affected. Similarly, daughter is affected, sons are also affected. So if this is the condition, we would automatically go and saying that it is X-linked dominant disorder. Right? So this is what we have. This is how, in simple, we start interpreting based on the simple rules what we have. Right? As I've already said yesterday in the previous session, right, that these rules have actually put forth by observing several pedigrees. Right? And it is actually speaking, it is not always correct, but to our knowledge, we can put it that. Right? So the condition here, this condition here tells us that this pedigree is an X-linked dominant pedigree. Right? Now let us start re-verifying it by putting the genotypes, by identifying the genotypes of each individual. Right? So when we say it is X-linked condition, we know that females have XX type of chromosomes and males have X and Y. So let us start putting it here. Right? So males have X and Y. Right? Males have X and Y. Females have X and X. Right? So since it is a dominant mutation, right? It is a dominant mutation. Let us not show anything here because X itself indicates that it is dominant mutation, right? Recessive mutation, a recessive condition is normal. So this condition would actually be normal, right? If this is the condition in case of females, the individual is said to be normal. If either this is the condition, one dominant and one recessive, that shows that it is a dominant condition. Therefore, the female individual will be affected. Okay, so let us start putting the genotypes here. Right? First one, the father will start giving which chromosomes to the females here? To the females, the father will give a dominant X chromosome. The father will give an X chromosome. Similarly, to this female also, the father will give X chromosome. Right? And to this male, the father will start giving Y chromosome. Y chromosome has come in. Now, what will the mother give? Since both are recessive alleles, right? So the mother will start giving X dot here and here also X dot here. Now, as we have said, it is a recessive mutation, right? It is a dominant mutation. So this is dominant and this is dominant. Right. So this is what we have here. Now, what type of thing will the female get here? Right. So the female will start getting, the male here will start getting X dot Y. So as we have said, this is a recessive condition here and this is a dominant. Now don't get confused. This is dominant and this is recessive, right? So in case of males here, it is X dot Y. That shows that this is a recessive condition. Since it is a recessive condition, the male is actually normal. Whereas in case of females, there is one dominant allele and one recessive allele. So it is said to be heterozygous dominant. Therefore, since it is a dominant disorder, the female is affected here. Here also, there is one dominant allele and one recessive allele. Right. Since there is a dominant and a recessive allele, this genotype would be heterozygous dominant again. Right. Since it is a dominant type of disorder, the female is affected. Now, don't get confused with that. Now, talking about this male here, they are telling that the male is normal. So the male will be X dot Y. And here also the male is normal. So the male will be X dot and Y. Right. Now, talking about the offsprings here in the third generation. Now, the male will start, all are males here. So the males will start getting Y chromosome from their father. Right. Males will all get Y chromosome from their father. And from the mother, which chromosome will come in? A dominant allele will start coming here. That's the reason why this child is affected. Whereas here, it would be a recessive allele which actually comes here. Right? So this would be the genotype of all the three children. One child is affected because of the dominant allele that is present on the X chromosome. Here, two children are unaffected because of the recessive alleles that are present in the X chromosomes. Similarly, in this condition, the father will start giving Y chromosome to the sons and the father will start giving X dot chromosomes to the daughters. That is the recessive chromosome to the daughters here. Right. Next, what would happen here? From the mother, since this child is not affected, the mother will start giving another X dot chromosome here. And here also another X dot chromosome here. Whereas to the male children, what will the mother give? Because both the children are affected, the mother will now start giving the dominant allele here. So X here and X. Therefore, this would be the genotypes of all of the individuals that are present in this particular X-linked dominant disorder. Right? So this is what we have with the third example, what we have with regards to the pedigree chart. So if this sort of a pedigree chart is given, right, we would confirm telling it that this particular disorder here is an X-linked dominant disorder. Right? So things what you need to remember, as I've already said, whenever a chart is given, pedigree chart is given, first identify whether it is dominant or recessive. That would be very simply done by observing all of the generation. If trait is present in all the generation, if affected individuals are present in all the generations, it would definitely be a dominant trait, right? If there is skipping of generation, it would be a recessive trait. Similarly, what else we saw next? How to identify whether it is autosomal or X-linked. So to identify whether it is autosomal, what do we look for? Affected father, affected son. That is one way how we identify autosomal. Second way how we identify autosomal, affected father has both affected son and daughter in the same generation. That is autosomal. X-linked condition, what did we say? Affected father has affected daughter. So that is the base rule what you need to remember with regards to X-linked condition. Affected father having affected daughter. That is again X-linked disorder. Another condition, what did I say? If, especially in case of recessive condition, if males are affected more than the females, then it would be X-linked recessive condition, right? So this is what we have an X-linked dominant disorder, right? I hope all are clear with this, right? So quickly take this down, then we'll move on to the next one. Okay, so let's take up with another example to identify the pedigree here, right? So let's take up example number 4 and see the pedigree here. Again, we have the same 4 things, same 4 options here. So from this, we need to identify which is what, right? So this would be pedigree number 4 here. Again, a 3-generation pedigree here. Right? A simple 3-generation pedigree here. So this would be generation 1, generation 2, and generation 3. So first, let us see what exactly is there here in this particular condition. A normal male and a normal female, they are married and they have 3 children. First daughter, second son, and the third again a daughter. The son is actually affected. Right. Now the third child marries a normal man and they have 2 children, of which both are said to be males, right? And both are said to be affected. Right? So what sort of a pedigree would this be? It is a simple 3-generation pedigree here. Now, as I've said, how do we go forth with it? First, identify whether it is dominant or recessive. By observing this, what we can make out, right? First generation, no one is affected. Second generation, 1 son is affected. Third generation, both the sons are affected. So, since there is skipping of generation, we would say that it is a recessive disorder. Right? So when we say recessive disorder, we are ruling out option number 1 and option number 3 here. So it could be either autosomal recessive or X-linked recessive. Now, how do we identify whether it is autosomal or X-linked? As I've already said again, I'm just repeating it once again, right? In case of autosomal, how do we identify whether it is autosomal? Affected father will have affected son. Is that condition seen here? Affected father, they are not having affected son. There is no proper information given here. So it cannot be autosomal here. Second condition, what did we say? Affected father has both affected son and daughter in the same generation. Is that possible here? That also is not seen here. Talking about X-linked condition. In X-linked condition, affected father will have affected daughter. Is that seen here? That is not seen here again. Right. Again, the third case here. Affected mother has affected son. Is that seen here? So that is also not seen. And I told you one more thing also. How do we identify whether it is X-linked recessive condition? If the number of males affected are said to be more than the number of females, then again we go with X-linked. So if you are not able to find out whether affected father has affected son or affected mother has affected daughter, or if you are not able to find out whether affected father has affected daughter or affected mother has affected son. If that is the case, we go in for again this. By looking at this, we can observe that males are actually more affected, whereas none of the females are affected. Now, since our males are actually more affected, we are already sure that it is recessive. Now, males are, since we are telling males are also more affected, then automatically we would go in for X-linked recessive condition. Right? We would go in for X-linked recessive condition here. Right? Now, let us again start re-verifying it by plotting the genotypes. Right? So, male here, we know male would be X and Y, normal male. So, it would be X and Y. Talking about female, there are two possibilities here. The female could be either XX or the female could be a carrier with one recessive allele being present, X.X. Right? So these are the two possibilities here. Now let us see with the offsprings. The father will start giving X chromosome to this child here. Right? The father will start giving Y chromosome to this son, and the father will start giving another X chromosome to this daughter here. Right? So these are the chromosomes which are given by the father. Now what chromosomes will the mother give here? Right. Since we are talking about a recessive condition, definitely the son is affected. So definitely a recessive gene should come to the son from the mother. So the mother will start giving X.Y. Now, since mother is giving X.Y, the genotype of the mother would be X.Y. Sorry, X.X. Now, to this child, the mother will give an X chromosome, or the mother could give X.X chromosome. So this is another possibility here. Here also the mother could give X dot to this, or the mother could give X and X. So these are the possible genotypes of the two children here. Now talking about this again, we are telling that in the second generation, the third child marries a normal man. So the normal man is X and Y. Since normal man is XY, father will give Y chromosome to their sons. And what should come from the mother here? Since both the children are affected, definitely the mother should give X dot, a recessive gene to the children. Now, since the mother is giving recessive gene, the genotype of the mother would be X dot X. Right? So this we are not sure because the further generations are not given. Right? So this we start as X-linked recessive condition or X-linked recessive mutation. Right? So this is how we start plotting the pedigree chart, and at the same time we start interpreting the pedigree chart. Right? In most of the cases in your exams, the pedigree chart will be given. So based on what I just said now, so by applying those, you can easily identify what sort of a genetic disorder it is. Whether it is autosomal dominant, autosomal recessive, X-linked dominant, or X-linked recessive. Apart from all of this, there is one more condition which is seen, which is called as Y-linked inheritance. Right? Y-linked inheritance. So what happens in Y-linked inheritance is the trait which is present in the father would be carried to the son. Right? In Y-linked inheritance, right? What are we talking about? Y-linked inheritance. Just like what we saw here as X-linked disorder, we have something called as Y-linked disorders also. Or rather, we can take it as conditions. We have Y-linked conditions also. So what happens in this Y-linked conditions is the genes that are present on the Y chromosome would get mutated. Now, since the genes present on the Y chromosome would get mutated, right? Whatever condition is seen in the father, the same condition will be seen even in case of children also. Right? So that sort of inheritance, we call it as Y-linked inheritance. Right? So if in a chart, right, if Y-linked inheritance is actually given, it would be that all of the males would be definitely affected. So if in one generation, if the male is affected here in the first generation, second generation also males will be affected. In the third generation also the males will be affected. So in a pedigree chart, if you see that all the males in all the generations are affected, then automatically we can say that it is Y-linked inheritance or Y-linked disorder, or rather Y-linked condition. Right? So this also sometimes you may get, but most frequently it would be these four options which are actually given. Fine. So this is what we have with pedigree analysis. So if a question is given, how do we plot a pedigree chart? That's what we have seen, right? And the second one, what we have seen is, if a pedigree chart is given, how do we interpret or how do we identify what sort of a disorder it is, right? So with this, we'll be done with our topic called as pedigree analysis, right? Fine. So let us begin with another topic, right? So since we are done with pedigree analysis, let us now begin with another topic, right, which is called as genetic disorders, right? So let us take up with the genetic disorders. So the topic what we'll be taking up now is genetic disorders. So what does this genetic disorders mean? What do you mean by genetic disorder? Gene related disorder or chromosome related disorder, we start calling it as genetic disorder, right? So if a particular disease is caused because of a mutation in a gene or because of a mutation in a chromosome, we start calling it as genetic disorder, right? So what exactly is a genetic disorder? If a disorder or a disease is caused because of a mutation in a gene, right, or because of a mutation in the chromosome, we start calling it as genetic disorder. So if you could recollect in the previous sessions, we spoke about a lot of things about mutations. We saw about different types of mutations. We classified all the mutations into two categories based on what is getting affected or what is getting mutated rather. Right. If chromosomes are getting mutated, we started calling it as chromosomal mutation. If you could recollect that, under chromosomal mutation, again, we saw two different types. One was called the structural mutation. If there is some sort of change in the structure of chromosome because of mutation, we call it as structural mutation. Second type under that was numeric mutation. If there is either increase in the number of chromosomes or rather decrease in the number of chromosomes, right, we started calling it as genomic mutation or numeric mutation. Similarly, with regards to gene mutation also we saw. So what did we say with regards to gene mutation? If mutation is seen in a particular gene on a chromosome, then we started calling it as gene mutation, right? So any disease or any disorder which is caused because of the mutation in the gene or because of mutation on the chromosomes, we start calling it as genetic disorders, right? So these genetic disorders was first identified by a scientist by name Sir Archibald Garrod. Right? So he was the first one who came up with this, who identified these genetic disorders, right? Sir Archibald Garrod. So he was a scientist who first identified this sort of a genetic disorder in the year 1908. Right? So in the year 1908, he came up with this sort of the genetic disorder. So the first genetic disorder that was identified by the scientist was termed as alkapton urea. It was termed as alkapton urea. So this is the first genetic disorder that was identified by the scientist. So it is considered as a metabolic disorder. It is considered as an inborn metabolic disorder or metabolic error. Inborn metabolic disorder. Right? So alkaptonuria was the first identified disorder, right, by a scientist by name Sir Archibald Garrod in the year 1908, right? And it is considered as an inborn metabolic disorder. So what does that mean? Inborn metabolic disorder? Inborn, what does inborn mean? Inborn means by birth. So by birth, a child is born with this sort of a disorder. Which sort of a disorder? Metabolic disorder. So what does that mean, metabolic disorder? Proper metabolism is not happening in this particular individual. So what exactly is this alkapton urea? Right. So in our body, right, there is one toxic compound that is actually formed in our body, which is called as homogentisic acid. Right. So this is actually said to be a highly toxic compound that is actually formed in the body. Homogentisic acid. In normal individuals, in normal individuals, this homogenesic acid goes to the liver, right? It goes to the liver, and in the liver, in the presence of an enzyme called homogenesis. So in the presence of this enzyme called homogenesis, this homogenesic acid would be transformed into a less toxic form, into a less toxic compound or less toxic form. This is actually the normal process that is happening in all of the individuals, right? So in all of the individuals in our body, we have this toxic compound called homogenesic acid that is being formed. This homogenesic acid goes to the liver, where the process of detoxification occurs. We know one of the major function of liver is undergoing a process called as detoxification, right? Converting the toxic substance into less toxic form. Similarly, this homogenesic acid, which is a highly toxic compound, once it goes to the liver, is being converted into less toxic form in the presence of an enzyme called homogenesis. So this homogenesis is also called as alkapton oxidase, right? It is also called as alkapton oxidase. So in the presence of this enzyme, homogenesic acid, a highly toxic compound, is being converted into a less toxic form. So this is in the normal human condition. But in these individuals who are suffering from this disorder called alkaptonuria, what exactly is happening is there is a mutation, right? Try to understand this. There is a mutation in a gene coding for the enzyme called as homogenesis, right? So what exactly is happening in individuals suffering from alkaptonuria? In these individuals, they have a mutation in the gene coding for an enzyme called homogenesis. Now, since this gene is mutated, what is happening? Homogenesis is not being synthesized, or homogenesis is not being formed. Now, since homogenesis is not being formed, what is happening? Homogenesic acid is not getting converted into a less toxic form. So, what would happen now? This homogenesic acid will start accumulating in the body. So, where all does this homogenesic acid accumulate? It starts accumulating in the cartilage, right? Number one. Then it starts accumulating in the joint capsule, right? At the joints where we have the joint capsule being present. It starts accumulating in the ligaments. It would start accumulating in the tendons. Now, because of this accumulation in all of these places, our skeletal system and the muscular system will not be able to work properly. It will not be able to function properly, right? So that is what we call it as alkaptonuria. Now, since the concentration of homogenesic acid increases in the body, some of the homogenesic acid will be excreted out through the urine. And this urine containing homogenesic acid, if it comes in contact with the air for some time, the urine will turn black in color. So it's also called as black urine disorder. Because why? The homogenesic acid will undergo the process of oxidation in the presence of air. As a result of which, the color of urine will start turning black. So therefore, they have called it as alkaptonuria. Right? So this was the first identified genetic disorder, which they call it as inborn metabolic disorder. So why do we call it as metabolic disorder? Because the proper metabolic activity is being affected here in this individual. And it is an inborn disorder, right? It is actually seen by birth. Therefore, we start calling it as alkaptonuria. So this is the disorder which was the first identified genetic disorder by Sir Archibald Garrod in the year 1908. Right? From then on, once this disorder was identified, several other genetic disorders have been identified. Right? So there are more than 200 different types of genetic disorders that have been identified, and all of those genetic disorders have been grouped into different classes. So we'll first take up with the classification, then we'll get into each of the genetic disorder in detail, right? So what exactly have we started off with? We have started off with the topic called as genetic disorder. So what exactly is this genetic disorder all about? The disorder that is caused due to mutation in the gene or mutation in the chromosome, we start calling as genetic disorder, right? So the genetic disorders was first identified or discovered or first identified by this scientist by name Sir Archibald Garrod in the year 1908. The first genetic disorder that was identified was alkaptonuria. So what is seen in what actually happens in alkaptonuria? It is an inborn metabolic disorder, right? It is an inborn metabolic disorder, right? So why it is said to be inborn? Because it is actually seen by birth. Why do we call it as metabolic disorder? Because the normal metabolic process is being affected. So what happens in this particular condition, right? In normal individuals, we have a highly toxic compound which is produced in our body, which is called as homogenesic acid. In normal individuals, the homogenesic acid will be transformed or converted into a less toxic form in the liver under the influence of an enzyme called as homogentisase or alkapton oxidase. Now, in these individuals suffering from this condition called alkaptonuria, what exactly is happening is there is a mutation in the gene, right, present on chromosome number 3, right? So this gene is actually present on chromosome number 3. So the gene that is present on chromosome number 3, coding for an enzyme called homogentisase is getting mutated. As a result of which homogentisase is not formed. Now, since homogentisase is not formed, homogentisic acid is not getting converted to less toxic. Therefore, this homogentisic acid will get accumulated in the body, right? And where all does it get accumulated? It gets accumulated and settles down in the, I give you a list of organs where it settles down. It settles down in the cartilage. It accumulates, it gets accumulated in the capsules, bone capsules or rather joint capsules. It gets accumulated in the ligaments. It gets accumulated in the tendons. And because of its higher concentration in the body, some amount of homogentisic acid is excreted through the urine. And this urine containing homogentisic acid, if it is allowed to come in contact with air for some time, it undergoes homogentisic acid, undergoes oxidation, and the urine will start turning black in color. Right? So therefore, we start calling it as black urine disorder, or it is also called as alkaptonuria. So this is the first identified genetic disorder by Sir Archibald Garrod. Right? So now let us take up the classification of genetic disorder. Then we'll get into each genetic disorder in detail. Right? So all the genetic disorders could be broadly classified into two categories. Right? So all genetic disorders could be broadly classified into two categories. So we start classifying them into two categories. Right? So the first one, the first category is called as gene disorders. The first category is called as gene disorders. Now, what does this gene disorders mean? This sort of a disorder is actually caused because of mutation in the genes. Right? So due to mutations in the gene, we start calling it as gene disorder. The second category, what we have here is called as chromosomal disorder. Chromosomal disorders. So what are chromosomal disorders? Any disorders which are seen due to mutations in the chromosome. We start calling it as chromosomal disorder. Now these gene disorders have different names. We start giving them different names. So it is also called as Mendelian disorder. Mendelian disorder. Why it is called Mendelian disorder? Because it actually follows the Mendel's inheritance pattern. Now, since it follows the Mendelian inheritance pattern, it is also called as Mendelian disorders, or it is also called as inherited disorders or inheritable disorders. Inheritable disorders. Whereas chromosomal disorders are called as non-Mendelian disorders. Non-Mendelian disorders, or we also call them as non-inheritable disorders. Non-inheritable disorders, right? Either non-Mendelian disorders or non-inheritable disorders. Now, what does this mean? Gene disorders are also called as heritable disorders. So, what does that mean? So, this sort of a mutation that is seen in the gene is inherited from one generation to the other generation. So, if a particular parents are suffering from this sort of a disorder, the same disorder would be seen even in the children also. So that sort of a disorder, we start calling it as inheritable disorder, inherited disorder, or Mendelian disorder, or gene disorder. Whereas chromosomal disorders are said to be non-inheritable disorders. So chromosomal disorders do not are not actually inherited from one generation to the other generation. So if in a particular generation, the individual is affected, that's it. The individual will not pass on that mutation to the next generation. So therefore, we start calling chromosomal disorders as non-inheritable disorders. This is what we have. Now, gene disorders could be in turn classified into two different categories. Right? So gene disorders could be in turn classified into two categories. So what are those two categories? First one would be autosomal disorder. Autosomal disorder. Autosomal gene disorder. And the other one would be X-linked gene disorder. Right? So it would be either autosomal gene disorder or X-linked gene disorder. Similarly, here also, the chromosomal disorders could be again classified into two categories. Same two categories here again. It would be autosomal chromosomal disorder or it would be X-linked chromosomal disorder. Now, what does this mean? Autosomal gene disorder means, so we know that in humans we have 22 pairs of autosomes. So, among this 22 pairs of autosomes, a mutation in any one gene among the 22 pairs of autosomes would be termed as autosomal gene disorders. Right? Then we have one pair of X chromosomes in case of females. So females have XX and males have X and Y. So if there is a mutation in this X chromosome, so those type of mutations or disorders we start calling as X-linked gene disorders. Similarly, here also with regard to chromosomal disorders here. In chromosomal disorders, what do we have? We saw that there are two different types of mutations, right? One is called structural mutation, other one is called numeric mutation. So if there is structural or numeric mutations happening in the autosomes, so we would start calling it as autosomal chromosomal disorder. Similarly with this also, X-linked. So if there is either increase or decrease in the X chromosomes or rather the allosomes, right? We start calling it as X-linked chromosomal disorder. Now again, this autosomal gene disorder would be again in turn classified into two categories. So the mutation here could be either a dominant mutation or it could be a recessive mutation. Right? It could be either a dominant autosomal mutation or it could be a recessive autosomal mutation. Right? So mutation caused due to autosomal genes or alleles, mutations caused due to recessive alleles. So here also, even in X-linked also, we would have the same condition here. So we would have dominant X-linked disorder, and at the same time we would have the recessive X-linked disorder. Right? So this is what we have with the classification, basic classification of the genetic disorder. So all genetic disorders fall into two categories, either gene disorders or chromosomal disorder. Gene disorders, we also call it as Mendelian disorder or inherited disorder or inheritable disorder. Right? So under gene disorder, we have two different classes again: autosomal gene disorder, X-linked gene disorder. Again, under autosomal gene disorder, we have dominant autosomal gene disorder or recessive autosomal gene disorder. Again, under X-linked, we have two different categories again. It could be either a dominant X-linked gene disorder or it could be a recessive X-linked gene disorder. Similarly, with chromosomal disorder, it is also referred to as non-Mendelian disorder or it is also called as non-inheritable disorders. So this chromosomal disorder again falls under two categories. One would be autosomal chromosomal disorder and the other one would be X-linked chromosomal disorder. So this is the basic classification of genetic disorders what we have, right? So from the next session onwards, we'll start taking up a couple of disorders under each of the category and we'll see what exactly is happening in those sort of disorders. Fine? Thank you.